Global Food Regulatory Science Society. GFORCE mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems. Consistent reliance on food regulatory sciences enables robust food control programs.
Good morning. Good morning. Is my voice heard?
Yes, good morning. Do you hear me?
Yes, we do, Dr. Philip.
Thank you.
Good morning, good evening, everyone, and welcome to today's webinar on the development and deployment of monitoring programs for hormones in food, with a focus on veterinary residues. My name is Dr. Riwa Gusous. I'm currently working as a food safety expert at Jordan Food and Drug Administration, JFDA. And I'm also a member of the Global Food Regulatory Science Society, GFORCE. I'm pleased to be today moderating today's webinar organized by GFORCE, and it's its third webinar as part of its Food Regulatory Science webinar series, and to have participants from all around the world today. Before I introduce our speaker and the topic, please note that Arabic interpretation is available for this webinar, and additionally, we encourage you to submit your questions throughout the presentation via the chat box. We will have a dedicated Q&A segment following Dr. Philip's talk to address your questions. As many of you know, the safety of food we consume is an issue of global importance. One area of concern is the presence of residues resulting from veterinary applications, including the possible use of hormones for that purpose. Controlling these residues involves the development of consistent and robust monitoring program to ensure that food we consume continues to be safe and also addresses consumers' preferences. Today, we will focus on these monitoring programs for veterinary residues, but we will focus more on hormones. We will explore how monitoring programs for veterinary residues are developed and deployed, and how they contribute to protecting consumers from potential health risks. And who better to talk about this topic than our speaker, Dr. Philip Delahu, a former director of the Laboratory of Hormonology in CER, Belgium, one of the pioneers of veterinary residues, testing and one that promoted the status of his laboratory to become a European reference center for testing of veterinary drugs in food. As I mentioned, Dr. Delouette has been a distinguished expert in veterinary medicine, residue detection, allergen detection, and animal experimentation. Dr. Philip will be walking us through the critical steps and challenges involved in developing and deploying effective monitoring programs for veterinary residues in food with emphasis on hormones. Again, thank you all for joining us today. And Dr. Philip, I am really privileged to be moderating this webinar where you are the presenter and you will be sharing your insights with us on this topic today. And with that, I'll hand it over to Dr. Philip.
Okay, just I have to.
You ready, Doctor?
Yes, I'm just to start. I just to put full screen. Okay. Do you see my screen?
Not yet.
Yes, we do now.
Yes.
I believe we do. Yes.
Yes. Okay. Thank you. Thank you, Huba, for this nice presentation.
It's my pleasure.
It's really a pleasure for me to share my experience regarding the problem of residue testing and particularly hormone. And I would like to thank Sami for inviting me today. During this presentation, after a small introduction, I propose to speak about the legislation in Europe. It's very important to have the support of a good legislation for such a control. And the main part of my presentation will be the development in analytical methods, with general consideration of the methods available for the moment. And after, I propose to discuss on some problems regarding the natural hormone, androgen, corticosteroid, and finally, beta agonists. And a small conclusion at the end of this talk.
That would be great. Thank you.
Thank you. So, why use anabolic in livestock production? It's first of all, anabolic and hormones are not the same thing. An anabolic is a substance that increases dietary nitrogen utilization. Most, but not all anabolic agents are hormone. It's important to remember that anabolic are mainly used in cattle because nitrogen conversion is poor. It's estimated that only 30% is converted in meat production. There is less interest in poultry and pigs where conversion is better. With anabolic, weight is increased about 10 to 20%. That's very interesting for the farmer and for the meat production. In the first synthetic substance used in 1938 was the diethylstilbestrol, but now it's been in all the country. And in Europe, all growth promoters are currently banned, and in some part of the world, some of them are partly legalized. Consistent legislation is important for control. Directive 96/22 states that the use of substance with an hormonal effect and antithyroid drugs and beta agonists are prohibited. But for other substance used in therapeutic, we are authorized to use, but in controlled condition.
Dr. Philip, is your camera on? Can you activate your camera also?
Just to help me. Could you help
If you
me?
want, Doctor, stop sharing, open your camera, then reshare again if you want. Thank you.
So I stop.
Stop share.
I close, okay?
Stop share in Zoom.
Okay, I stop. Okay. And okay. Sorry, excuse me.
No, because we want to see your face.
No, okay. And now I have to start again. Partager, okay.
No worries.
No, it's just a presentation I have to share. Do you see my screen now? Do you see is it okay now?
Yes, very good.
Yeah. Okay, sorry.
Oui.
Okay, I just. Okay, so some substances are allowed, but with a very strict control. It's very important to treat the animals. The next regulation, it's 22/1644. It describes the requirement for the substance use in some case. But first, in the Annex A, we have a list of all the substances that are forbidden. And we also have to add some substance regarding another regulation, is the regulation on MRL. I will explain a little bit later. And in this regulation, chloramphenicol, dimetrazole, and other substances are forbidden. So we are not allowed to use this product. Chloramphenicol is a well-known antibiotic, very often used previously, but now it's totally forbidden. There is also in this group the phenylbutazone. Regarding the number sample that we have to take during the control, we have now, since a new directive in 22, two types of plan. The first one is the responsibility of each national country and is based on risk-based national. So you understand very easy that the problem is not the same in France than in Denmark. In France, it will be more focused on cattle, on beef production, and in Denmark is the fish production more important than in other countries. There is a second plan, is what we call EU monitoring. Europe imposed all the country a certain number of sample. For example, is 195 that we are obliged to do every year. And the third plan is for the import of the food in different countries. So each country have to evaluate the risk during the importation of foodstuff. The maximum residue level is also another very important concept. We have set up maximum residue level because we are authorized to treat the animal, but the residue must be below a certain level. And this level is based on toxicological data and food consumption on each country. Each substance has a level that we are not allowed to be over. I will show you an example in the next slide. The next directive and the 37 here give a list of all the pharmacological substance authorized in Europe with the maximum residue level. So the producer of drugs, the veterinary industry, before to launch a new product on the market, must make some experience in order to determine the maximum residue limit. So usually we treat a certain number of animals with a drug, and we calculate the withdrawal time before to be under the MRL. And this slide gives you the example for forphenicol. So the active compound is forphenicol. Forphenicol is an antibiotic who replaces now chloramphenicol. And the second column gives you the metabolite or the product that we have to follow to measure, to estimate the MRL. In the case of 4-phenicol, we have to determine the quantity of 4-phenicol is the parent drug, and also some metabolic. In this case, it's 4-phenycolamine. And the third column, you have all the species for which this antibiotic is allowed. And here it's the level for the different tissues. For example, for bovine, ovine, and caprine, you are allowed to use 4-phenicol if the MRL is below 200 for muscle, 3,000 for liver, kidney, and so on. So you have the data for all the different species. In some case, the product is allowed only for 1 or 2 species, but in this case, it's very large use in animals. And as you can see on this column, it's indicate that it's not allowed for milk producer animals. Yeah, it's normal because we are not allowed to have antibiotics in milk. So there is a restriction for animals who are producing milk. The next directive, the decision of Commission of 02, give the performance of the analytical methods. So we have to follow before to launch an analytical method for the detection of residues, all the criteria necessary to could interpret the result. Globally, there is 2 type of method. The screening methods, I will give you more information about that later. It's mainly ELISA. And for the screening method, you are allowed to have false positive result. It's not a problem if you have too much positive result, because a screening method must be confirmed by a confirmatory method. But for the screening method, you are not allowed to have false negative results. And the screening method and the confirmation method, give you exact information of the chemical structure of the analyte. Most of the time is LC-MS used for that purpose. The first thing before to start to develop a method is to know which is the target molecule and the metabolite of this molecule. It's very important, it's essential to know the target molecule and the metabolite, because you must know what you search in your foodstuff. Most of the time, the molecule is itself in the foodstuff, but in some case, the parent drug is no more present and you have the metabolite. The second thing is to know which matrix we have to control. For forbidden product, it's not necessary to have the animal product itself, like milk, meat, offal. Most of the time we use urine. Why? Because urine is the most root extraction of most drug and easy to collect at the slaughterhouse or at the farm. But for substances with MRL, with maximum residue limit, we have to take the edible tissues, because you have to check in this tissue if we are above or below the MRL. Some case for forbidden substance, we use other matrix. I think to the ester, we have the possibility to see in air, but it's not very often used. And the progestagen are in fat. They are concentrated in fat. So to control the misuse of progestagen, we use the matrix fat. In the analysis step, we have several steps. We need clearly to be identified. First, we have the sampling. And the sampling is important if we want to obtain a representative result. It must respond to the request and the representative. There is no problem for urine sample, but it's more complicated for meat or other solid sample. And the sampling we had usually for the confirmatory method like LCMS, an internal standard. The internal standard is necessary for quantification because during all the steps of the analysis, we lose substance, we lose a part of the molecule. So if you need exact quantification, it's necessary to calculate the recovery, which quantity you have at the end of all the steps. The next step for urine sample is the hydrolysis, because urine is excreted on conjugate form, and first we have to perform the deconjugation. Most of the time it's performed by using enzyme like helix pomacia. There is two forms of conjugate, glucurono and sulfur. After this step, we have the extraction. So we have to extract from the matrix the compound of interest. But it's not a specific extraction. Usually we use liquid extraction with organic solvent. As it's not a very specific extraction, we have to perform a purification. This purification will be done most of the time on solid phase column like C18. But in the next slide, I will show you some very nice and very pure technique, very nice technique on immunoaffinity chromatography. It's used for some purpose in residue analysis. And the two next steps are the HPLC-MS. HPLC have for objective to separate the different different compounds before identification by mass spectrometry. And finally, it's necessary to perform the validation of all this process in order to have good results. I show you the principle for the immunoaffinity chromatography. So it's the first thing before to perform such a purification is necessary to have antibodies. This technique is also used in the field of mycotoxin. You couple your antibodies to cephalode beads, and on the top of the column, you had your liquid sample. You have a binding between here. It's also possible to couple several antibodies. It depends what we would like to detect. And in the next step, you have a binding of your compound of interest with the antibody. And we perform some washing step in order to remove the interference. So you have a very clear, you have a very pure sample with only the compound of interest who are bound to the antibodies. And the third step at elution of your compound before a quantitative determination. We have used this technique for the purification of several androgen, estrogen, beta agonists, and corticosteroid in my lab by coupling the cephalose bead with, let's say, if I remember well, 7 or 10 different antibodies. And so you have a very clear extract with only the compound of interest. All the interference are removed. In this case, you have a specific purification. We also have produced for the human doping lab control in Cologne, a system for the purification of different peptides used in human doping. So as you can see, we have here antibodies against insulin, LHRH, ACTH, GHRH, and IGF-1. All these antibodies are in a gel, and we had in this gel second antibodies, and we couple two magnetic beads, and we have a link with these second antibodies with the first one, and it's very easy to purify the sample by using a magnet. So by this way, you have a very clean chromatogram, and it's easy to detect all these peptides used in human doping. For example, we had a nice application with insulin. As you know, insulin is used in cycling to have glucose disponible, and but there is natural insulin and xenobiotic insulin. So it's important to make the difference between the different sort of insulin, and by using these antibodies, we are able on mass spectrometry to make this difference. It's used in routing in human doping. Before to start with a technique, it's important to know exactly what we need. And there is two types of method. Rapid method in immunoassay, most of the time is immunoassay, like ELISA or lateral flow device, or mass spectrometry method, LC-MS. The characteristic of each method of these both methods are totally different. The first one is available in the field and mainly the lateral flow device. This technique must be rapid. As explained previously, no false positive result, no extraction step and sheet. On the other hand, mass spectrometry are laboratory technique. You need very special and very expensive equipment. They are specific. You have an ID of your molecule. You are not allowed to have false positive results. And most of the time, you have some very difficult and very complex extraction step. And the later point is that techniques are expensive. So this one are mainly used on the field, and it's important for, let's say, the food producer to know if it's okay or not. But if it's not okay, he must confirm by a mass spectrometry technique. In this slide, you have the principle of ELISA. So you have 4 steps. In the first step, we coated on a plate. It's indirect. In this case, I explained to you indirect ELISA. You coat, let's say, a non-specific antibody. It's what we call a double antibody. It's an anti-IgG antibody. Most of the time, it's anti-IgG of rabbit, because the antibodies, again, all the steroids and so on, I produce in rabbit by polyclonal technique. On the second step, you had the specific antibody of the compound that you are interested to detect, and you have a binding between the second antibody and the first antibody, the capture antibody. On the third step, you had in competition the target antigen if it's present in the sample, and the target antigen coupled to an enzyme, to HRP or radice peroxidase. So you have here a label peroxidase, and you have a competition between this target antigen and the antigen conjugated to an enzyme. And we had a TMB in order to have a change of color. And as you can imagine, it's inverse proportional, the color, it's inverse proportional to the concentration of the target antigen. If you have a lot of target antigen, you have a very small quantity of color of antigen coupled to the HRP. We can, after that, we can do a standard curve by increased quantity of no antigen. Usually we start from null, okay, to 1,000, 10,000. And on the other axis, we put the signal, the intensity of the color of your wells. Well, as you can see, if you have a small quantity of antigen in your sample, you have a high signal. In other hand, if you have a quite high quantity, you have a very low signal, is due to the competition between free and label antigen. And we have a standard curve in order to perform a quantification of your sample. There is an important point is the relative signal F50 here, and it's what we call the inflection point of your curve. And it's important to make a good quantification to have 6 points on the linear part of your curve. On the two other parts, this one and this one is difficult to have, it's impossible to have a good quantification. The other system used on the field is the lateral flow device. And it's particularly interesting for the simplicity, speed, and cost effectiveness. When a sample is introduced here in the sample path, so we have like a chromatography column on a nitrocellulose membrane, the sample moves from this part to the other part. Here on the conjugate part, you have an antibody. In this case, it's a specific antibody, again, antigen, coupled to a detector system based on gold. And you have a conjugation here between the residue, the target molecule, and the antibody. And you have here on the membrane or migration of your sample and a first test zone with couple of your first antibody to a second antibody. And finally, you have a control zone and the absorption path. And if you have the presence of antigen in your sample, you have a color here and a small color on the control zone. For positive sample, you must have two colors. For negative sample, only one here. This system is very interesting for rapid control and rapid test on the field. That's the two major systems used for the screening methods. I cannot resist to show you but development that we have performed during an European project called BIACOR with a group of Belfast and Wageningen in Netherlands. It's the principle of the surface plasmon resonance is the biosensor system produced by BIACOR. We have a binding of biomolecule on the surface of the on the surface. So in the system, we have three parts. The first one is a flow channel here, and we code here antibodies, specific antibodies, again, antigen. The second part is a surface with a gold fill, and the third part is a light source with an optical detection unit. When you inject your sample, if you have antigen, if you have drugs in your sample, you have a bond with the specific antibody. And your light is, you have a shift of your light. If there is no antigen, no residues, no drugs, we have the angle 1, you have the shift here, 1. And if you have some substance present in the flow channel, you have a shift of your reflected light. So in the system, we measure the shift, and the shift is proportional to the quantity of antigen of drug present in the flow channel. We have a very nice project, but it's not often used because it's quite complicated system to use in routine in the lab. But there is some other application for this system in human medicine. Another project that we have developed with people of Netherlands is the multiplexing immunoassay. All the systems I show you are based on antibody-antigen reaction. And here we use different microsphere. You have possibility to have 3 microspheres of different size. I think that here we show 2, 1, and a bigger one here. And you have the possibility to use 4 different antibodies. So by this multiplexing, you can check 12 compounds. And you have the possibility to have the direct system. The antigen is coded to the bead, and we are coming with a specific antibody and a label of fluorochrome. And in the other system is like indirect system for ELISA. We have the bead, we have the antigen, we have we have a primary antibody, and the second antibody in this case is labeled to fluorochrome. The advantage of this system is that you need only 1 secondary antibody for all the beads and for all the antibody. Now, the confirmatory, most of the confirmatory method use LC-MS since, let's say, 20 years. And LC-MS is the technique of choice for all labs involved in the control. You have different parts. The first part is the liquid chromatography with the compound with mobile phase is usually 2 different solvents, and you have the possibility to adapt the concentration of each solvent. The second part, you have the LC column, and here we have the system of injection. So we inject the sample in the column, and we have a separation of different peaks regarding the mobility on the LC column. After that, we have a ionization chamber, and we have the first mass analysis, and all the ion, the first ion, let's say, is the parent ion, is characterized by a mass to charge difference. After this first mass analyzer, we have a collision chamber with inert gas, and we have a second system to analyze the mass, and we have the detector. We give you the mass to charge for each compound. The mass to charge with the parent ion and the daughter ion are what we call the ID of the molecule. This technique is used in most of the labo, and here I describe you the system solved by SciEx and Waters. Another system interesting to evaluate the possibility of endogen of exogen, because as I would explain later, it's difficult to say, for example, is endogen testosterone or exogen testosterone, is what we call IRMS, isotope ratio mass spectrometry. And in this case, we use the ratio of isotopes, say, 1, 3, and 12, to know the origin of the steroid excreted. and it's interesting for metabolite in animals and feed mainly for androgen hormone. But in some case, you can solve some problem. I will explain later with some application, mainly for the androgen. This slide is just to explain to you how we can do the difference between artificial and human steroid. You have to know that the human steroid are mainly from C4 plant and the carbon dioxide for air. But for artificial steroid, they are mainly coming for C3 plant like soy, and that's the reason why we have different carbon ratio between human and artificial steroid, even if the chemical structure is the same. So, in the first part of this system, you have a combustion of few samples. The sample can be liquid, gas, or solid. And he converted the sample in CO2, NH2, and H2O. The next step, you have to remove the water. And after, we have the mass separation here with the ion accelerator and the electro trap. And you have a separation with also the ratio to mass to 44, 45, and 46. It's very important for such a system to have very clean peaks. Otherwise, you are not able to see the difference for the different mass ratio. Okay, but all this system have a cost. For example, this system costs around 200,000, and with all the peripherals, the price is close to 300,000. For all the people who would like to more information on this technique, it's you can take contact with Thomas Pieper at the University of Cologne Institute of Biochemistry. He has developed a lot of methods for the control of natural hormone in human doping. So he has a very large experience in this field, and there is no problem to contact Thomas. Now that we know that the two main techniques used for the control of residues are or qualitative or quantitative, what are the criteria for the validation of these methods? For the qualitative method, it's possible to have screening method or confirmatory method, and the same for quantitative method. The first thing that we have to check is the presence. It's what we call CC beta, detection of the substance. Is the substance, is the drug present or not? So for all the method, qualitative and quantitative screening and confirmatory, that's the first point that we have to prove. After that, we have what we call the decision limit. The decision limit is an important factor for all the confirmatory methods. So you have to set up your decision, you have to define your decision method or for qualitative and also for quantitative method, but in case on confirmatory. The trueness and the recovery, it's important for LCMS method. As explained previously, we lose the molecule during all the process of purification, and to have a good quantification is necessary to calculate your recovery before to give an exact quantity present in your sample. Precision is to determine for quantitative method, and selectivity and specificity for all methods. You have to prove that the compound that you are interested to detect is really this compound. And the last point to to validate it is the stability, the applicability. For me, that's very important, let's say, for qualitative method. Why? Because these methods are used on the field. And you know that, okay, the condition in a solid country, the temperature condition are not the same in all the country. Okay, now I propose to go a little bit more in details regarding the different group of hormone used for the anabolization of animals. Steroid hormones are synthesized from cholesterol during the steroidogenesis. All the different hormones are coming from the cholesterol. And this figure shows the molecular structure, and metabolic pathway of various steroids. Steroid hormones are synthesized in various sites, such as cortex of adrenal gland, gonad, testis and ovary, and placenta. But there is also some extraglandular production. Steroids play a critical role in regulation of water, homeostasis, salt homeostasis, metabolism, stress response for corticosteroid, initiation and maintenance of sexual differentiation and reproduction. They are classified in 4 groups. Estrogen are reproductive hormone in female, and the major compound is estradiol. Androgen are reproductive hormone in male, with the major compound, the most important is testosterone. Progesterone, progestogen are compound of pregnancy, and progesterone maintain the pregnancy in most of the animals. And finally, corticosteroids are the stress hormone with the natural cortisol eliminated on the form of cortisol. All these steroids are eliminated in urine on glucuronide or sulfate form. So, as explained previously, it's important to know the metabolism of the different compounds. Compounds are metabolized, and let's say for the steroid, we have a lot of different metabolites. It's not the same for antibiotics or corticosteroid, but in case of hormone, steroid hormone, we have to identify the metabolite. And these compounds are, most of the steroids are administered on beta form. So, sorry, we have, let's say, I take testosterone, we produce an ester on this part of the molecule. It's the 17 atoms, and we can couple very easy a side chain. It's what we call ester, and we found the ester on the ejection site. The objective of esterification is to allow a slow release of the compound. In some cases, for example, for nandrolone, we have decanoate form, and we have a release during several days, maybe 5-7 days. Otherwise, if you inject your molecule, the parent drug without esterification, after 6 hours, we have no more active product on the injection site. For most of the steroids, the first step of metabolization is an epimerization from beta to alpha form, is again on this site. So the OH now is in beta, and it's transformed to alpha form. The first phase of the metabolism is introduce polar group like oxidation, hydroxylation, and reduction, and it's done in the liver with this cytochrome 450. And the second phase is done in the kidney, as explained by glucuronic or sulfate conjugation. Several substances are allowed to use in some country, and that's the reason why it's important to have a maximum residue. For example, trembolone is used in US and in South America, and so but it's allowed under certain condition. And in case of trembolone, the MRL is 2 ppb for beta trembolone in muscle, and in liver is 10 ppb for alpha trembolone. As you can see, you have a pyramidization product with alpha form. For the natural compound, it's difficult to give an MRL because it's present in all animals and it's depend of the cycles, if it's young of adult bull, young of old cow, the level of testosterone, estradiol, or progesterone are not the same. There is also a progestagen called menagestrol acetate authorized in some country, and here you have the data for the MRL. So you are allowed to use in US MGA, menagestrol acetate, but you have to keep in mind that you must be below this level. And the same for zeranol. Now, I propose to see with you some problem that we have to face with some substances. And the problem is due to the occurrence of a compound that we call, let's say, semi-natural. So they are not allowed, but in some case, we find this compound in the sample with some specific situation, like androgen production, pregnancy, stress, and so on. You have to know that now with the new, the modern analytical technique, we are able to go lower and lower. We are very, very sensitive. It's not a problem now to detect a substance at ppb level, and in some case in ppt level. Otherwise, it's very easy to find some interference coming from natural metabolism of some substance. And I propose in the next slide to speak about the problem of thioracil, of natural hormone, 7-beta estradiol, testosterone, and progesterone, 19-or testosterone, boldenone, and corticosteroid. For all the natural hormones, you have variation according sex, yes, estradiol, testosterone, male, female, female, male, age, young, adult, season, and some pathological condition. You have to know that in male, it's 4-6 months, we have level close to 1 to 4 ppb, and adult bulls, it's around 10 ppb. But one of the problems of testosterone is that you have nictemeral variation. So you have variation during the day. Sometimes during the day, you have level of 1 ppb, and 1 hour later, you have 10 ppb. So you are very careful when you analyze the secretion of testosterone in male to put a lot of sample during the day. And something very special that we have seen that the plasma level in a treated animal with testosterone is lower than in non-treated animal. Why? Because we have a negative feedback from pituitary gland. So we have all a mechanism of feedback. When there is too much testosterone, we have a feedback negative. And most of the derivatives of testosterone are injected on ester form with benzoate, propionate, and nathionate. The 17-beta estradiol is very low in adult cow. So during the estrus cow, we have during the estrus phase, it's the phase when the cow is in heat, we have level of 0.01. So it's at the PPT level. And also in this case, it's injected on benzoate and cypionate form. Progesterone is the hormone of pregnancy, and the level, we have a variation between 0 to 10 ppb. But progesterone, most of the time, is injected in association with estrogen and androgen in order to calm the sexual aspect. Otherwise, when we have several cows together inject with estrogen or male inject with androgen, you have a lot of problems in the same room. Here you have the chemical structure of the different natural hormone. Here the testosterone, and you see that the difference with estrogen is only on the A ring. You have 3 double bonds instead of 1. And here with progesterone is the same as testosterone, except that we have position 17, a side chain with a total 21 atoms of carbon. What's the analytical method for natural hormone? The first one and the old one was to see the modification, the histological modification on the prostate for male V calf. Okay, it's easy, very old technique, but not specific, and it's only for male treated with estrogenic substance. We have a lot of immunoassays, mainly area. All this area were developed to evaluate the endocrinology in animals. So let's say in the 70, in veterinary medicine, we have set up a lot of area to know the endocrinology of the different species, but mainly horses and cow. And all this area are at PBB and PPT level. Confirmatory methods. So use the steroid ester, and it's very easy to confirm by LCMS, but we have to collect at slaughterhouse. So when the expert see at the slaughterhouse an injection site, or think that it's an injection site, you have to collect this injection site, and we have usually high quantity of steroid ester. We also have the possibility to detect in air sample, but it's not so easy because the extraction is very complicated and we risk an environmental contamination. And the method of choice is isotope ratio mass spectrometry. And in this case, you are able to make the difference between endogenous and synthetic form. This technique is used in human doping. And I don't know if you remember at the Tour de France, Flo Landis was found positive for testosterone with 0.6. Here you have the chemical structure of all the estrogen. So you have the natural estrogen, in this case, estrone, estradiol with 2 diol, 2 OH group, and estriol with 3 OH group. We have some synthetic estrogen. Ethylene estradiol is used in pill in association with progestagen. And we have here the stilbene. I will give some word in the next slide on this group of compound. And finally, the zeranol. Zeranol is extract from mold of Gibberella zeae. And we have, let's say, not very important estrogenic effect, but its use is interesting for fattening the cattle. As explained in the first slides, the stilbene was the first synthetic hormone used, and mainly in poultry. Now it's banned in all countries because we have detected some carcinogenic effect and it classified in ERC in group B2. It is created in urine or conjugate form, and the detection is very easy by immunoassay and mass spectrometry. So here you have the structure. You have two phenol groups on all the three, but you have some modification here in the middle of between the two phenol groups. For the detection of estrogen, we have developed, let's say, some radioimmunoassay or ELISA. For the radioimmunoassay, we have produced a polyclonal antibody by coupling BSA, bovine serum albumin, on this part of the molecule. And the radiolabel was tritium, and it's by added 2 tritium atom on this part of the molecule. And for the ELISA, the conjugate is done by deazotation on the NH on this part of the molecule. The immunoassay was very interesting, and the positive impact of the immunoassay for DES is that this is no more longer used in animal production because it was a very performance test. The next group is the androgen with testosterone. Testosterone is the natural androgen, but we have a lot of different androgen used, and these compounds are a lot of use in the field of bodybuilder. Mainly boldenone and methylboldenone are very often used, and also nortestosterone. So you see the difference are not important, and I propose to discuss a little bit later with you about trembolone, nortestosterone, boldenone, and stanozolol. Alors, trembolone is a molecule developed by the group Russell-Uclaf, and it authorized a large use in some countries like US. It's a very interesting anabolic agent and with androgenic action. And as you can see, it's injected on ester form with an acetate group, and it's used in implant form. So we put on the base of E, and most of the time is in combination with the etradiol. This molecule is very specific and very difficult to produce for organic chemistry because you have 3 double bonds on ring A, B, and C. And for organic synthesis, it's quite complicated. But this molecule was produced by Russell-Uclaf, and it's Jouquet and Salmon in France. We have produced this molecule, I think, at the 70, 80, something like that. It's metabolized in A form, mainly in urine, and in alpha form. Sorry, it's metabolized. No, sorry, it's a mistake here. In beta form, in alpha form. No, it's correct. And urine and in feces. So the detection, for the detection, it's important to have an antibody who is able to recognize alpha form, and by LCMS, it's not a problem. It's the 2 major techniques used for the detection of trembolone. But its large use is probably the most important product in fattening cattle. Everybody remember the Olympic Games in Seoul, and so the winner of this 100 meter is Johnson, but 48 years later, after his victory, he was disqualified for doping, and the substance he used was stanozolol. This slide is important to understand the detection of stanozolol. The structure of stanozolol is exact the same as methyltestosterone, except that we have a pyrazole group on the A ring of the methyltestosterone molecule. There is 3 metabolites for stanozolol, the 3' hydroxy here, and the 4-beta here on A ring. That's the two main metabolites in human. In cattle, the metabolite is the 16-beta hydroxy. So the system for detection of stanozolol in human and in cattle is totally different. The stanozolol is, as explained, used in animal production, and the metabolites are different. So for the detection by ELISA in bovine urine, we have developed an antibody, again, 17. So we have coupled here the BSA, the carrier, in order to mask this position of the molecule, and the same with the enzyme HRP, horseradish peroxidase. And by this system, we have a cross-reactivity of 100% for stanozolol itself and the metabolite. So this system is very good for the detection of stanozolol in bovine urine, but it doesn't work for human doping. And the detection is performed by GCMS previously and now by LCMS, and it's very easy to detect the stanozolol and the 3 metabolite by LCMS. Another compound very often used is nortestosterone. Nortestosterone, called also nordrolone, is used in veterinary medicine and is still allowed for some purpose, but not for fattening. And for nortestosterone, we have a lot of different ester, and the objective of the ester is to have a slow release of the compound. And one very interesting is the low rat, because it's a very long side chain, and we have a very slow release. It's also metabolized on alpha form, and we have a detection by ELISA or by LCMS. The problem with nortestosterone, that 17-beta nortestosterone, is endogenous in boar and stallion, in horse and pig. And it's also endogenous in pregnant cow and just newborn calf. Another androgen with some problem is boldenone. There is very few difference between boldenone and testosterone. You see exactly the same, except a double bond here for boldenone on A ring, between 1 and 2. The precursor of boldenone and testosterone are ADD and AED, as explained here. It's used on ester form, and for the detection, we use LCMS. In urine of animal treated with boldenone, we have a lot of different metabolites. But the problem is to make the difference between treated and untreated. So for pigs, we see we have 17-beta boldenone in male. Cattle is alpha, as explained, in newborn calf and cow and pregnant cow, and in horse, it's 17-beta present. We think that the difference, so the modification on this part of the molecule between boldenone is due to an enzyme called steroid dehydrogenase, and maybe it's coming from phase. And by this enzyme, so it's natural production of boldenone in pigs and cattle. For horse and pigs, for cattle. For horse and pigs, we see that 17-beta boldenone is produced in testes of male by aromatization of estrogen. Is a well-known way of transformation. So you see that it's possible to have from testosterone to have a natural production of boldenone. The progesterone, there is a lot of different progestagen. Some of them are authorized like megestrol. Medroxyprogesterone is also used in therapeutic for small animals, but megestrol, melangestrol, and chlorbanidone are sometimes used for the regulation of the cyclus in animals. But progesterone is an hormone produced by the corpus luteus, and it's secreted for maintain the gestation. Corticosteroid was a problem 10 years, 20 years ago in Europe because we have found a lot of positive results in urine, mainly of pigs. And corticosteroid, we have the cortisol, is the natural hormone produced by the corticosurrenal, by the adrenal glands. It's what we call the hormone of stress. So you have variation during the day, but it's mainly depend of the stress. And you see that the difference between prednisolone, with that drug used, very often used for inflammatory and autoimmune problems, there is only a double band of difference. And we think that the question that the transformation is the same as for boldenone. So it's a steroid dehydrogenase who modified the molecule and allowed to have a double bond on the arin. So we have, as explained, found prednisolone, so the synthetic corticosteroid, in porcine, equine, and bovine urine sample, and it's mainly after stress. Due to transport in pigs, it's very often we have a lot of stress before to be slaughtered, or you can induce artificially by injection of acetylase, substance like synactin. And when you inject synactin, you have an increase of cortisol, but also formation of prednisolone. And in Europe, we consider the following threshold level. We consider the level of 5 micrograms to be compliance of suspect sample. Another interesting ratio is the ratio prednisolone-cortisol in liver. But as you can see, it's possible to have a transformation of cortisol to prednisolone due to some enzyme. And that's the fact that we have to be careful when we detect prednisolone in urine sample. It can come in from cortisone after a stress. The next compound I would like to speak, it's the thioracil. The thioracil and thyreostat, we have a lot of molecules in this group like thioracil, tapazole, methylthioracil, are active per host and increase the weight. But it's not a production of protein. It's mainly due to increase in water absorption and retention. So you increase the quantity of water, not of meat. And this substance produces an inhibiting of the thyroid hormone production. It's a gland based on the base of the neck. And it's also a possibility carcinogen. Here you have a chemical structure of thioracil. They have been in Europe since a long time. And we estimated that it's necessary to have 5 grams per day to reach a weight gain. So it's a quite high, it's necessary to have a quite high quantity to have some increase of water. And the method, there is also a non-specific method is to weigh the thyroid gland. The gland is increased in large proportion, but okay, it's not specific. And you see all treatment. You have the HPTLC. It was developed, I performed singular chromatography. This technique was developed by Hubert de Brabander at Ghent University, but now we have the LCMS and the ERMS to make the difference between the ratio of carbon. But with thioracil, we also have some problem from some substance in the feed. The cruciferous brassica vegetable contains substances that are modified the intake of thyroid gland. And it's what we call hypothyroidism. So that's the reason why we have also put a threshold level. As explained, you need a big quantity to have an effect in the cattle on the way. But in case of vegetable in the feed, we have low concentration of thiouresil in urine. And so we have put a threshold level at 10 in cattle and 30 in pig urine. If there is some problem for the detection, if you are not sure, we can use some metabolite. So 6-methylthiouresil was identified as indicator of thiouresil abuse, where 4-thiouresil of endogenous production. So we have first to detect the thiouresil and with a level of 10 of 30, and after we have to check the two metabolites. The first one is thiouresil abuse, and the second one endogenous production. So is the flow chart decision, so 10, 30, and after, so is the 4 thiouresil metabolite is present, or yes or no, yes, it's confirmed, it's not present, it's non-confirmed, and the same for the pig. That's the flow chart used for the decision of use of thiouresil in pigs and cattle. I would like to finish my talk with the beta agonists. In fact, the beta agonists are not hormone. It's a structure similar to catecholamine, like adrenaline, and they are used in animals for respiratory disorder. It's very often used in horses, but we can also use this compound for parturition in cow. And in fact, it's also doesn't increase the intake of nitrogen, the transformation of nitrogen in a protein, but the beta agonists decrease the quantity of fat, and by this way, they increase the quantity of muscle. These substances are active per os and eliminated in urine and feces without being metabolized. So it's easy to detect the beta agonists. They are not metabolized. You have to detect the product itself. Ractopamine and zilpaterol are authorized in some countries for pig production, but there was some problem with misdosing of clenbuterol after consumption in liver. It was in Spain in 89 and 92. There was some intoxication with people who have eaten some meat, liver from cow treated with clenbuterol, and the symptoms are tachycardia, palpitation, twitching, and the detection can be performed by immunoassay and LCMs. Here you have the structure of most of the beta agonists. Clenbuterol is the most often used. You have amine group here, and this group is common at a lot of beta agonists. You have an aromatic change with some modification. Here, the ractopamine is quite different because we have 2 aromatic change, but all these 5 beta agonists are quite similar, and the zilpaterol is totally different from chemical structure. It was possible with different antibody and different tracer to produce generic immunoassay. The objective was to detect the maximum beta agonists in the same run. And you see here, there was different system. We have 2 areas, and all the 4 tests here are ELISA. And the first one is clenbuterol antibody and tracer clenbuterol. And you see it's a specific clenbuterol test. And you see that we have good cross-reactivity for clenbuterol, but not for salbutamol, malbuterol, simaterol is very good also. The 2 major beta agonists are clenbuterol and salbutamol. The second one is a mix of salbutamol and clenbuterol, and you see that in this case we improve the cross-reactivity. We have used during a very long time this test in our lab, and also in Holland. The next step are ELISA, because we have replaced the radioactive by ELISA, and you have 4 different tests. The first one is specific for clenbuterol, and you see again, we have poor cross-reactivity with salbutamol. For most of the other is quite good. The D test is a mix of salbutamol and conjugate clenbuterol. It's better for salbutamol, malbuterol, simaterol. And this one was also large use, and most of the company proposed this test for the moment. And it's a mix of antibody. There is 2 antibody, one against clenbuterol and the other one against salbutamol with a clenbuterol conjugate. And the last one is a specific salbutamol. So you see by combination, some kits by combination antibody and clenbuterol is possible to manage the cross-reactivity of the different beta agonists. For my conclusion, I would like to say that before to start control, it's important to analyze all the toxicological data. There is a lot of international organization. There is a lot of data. I have shown you some of them regarding the authorized product, but you have plenty of information on that point. The second thing is to ask the regulatory agency to make a risk analysis. As explained, in Europe, there is a ban for all the compounds. In US, some of them are authorized, in South America also. I will not say if it's better to authorize or not. Let's say in the case of Europe, there is also a political reason. I have not to give an advice about that, but there is political reason to ban all the substance, but the situation is not the same in US. And for the implementation of the control, it's a very complex. You have first to know where you perform the control. Is it on the slaughterhouse? It's to the food producer. The second point to evaluate is the matrix of interest, number of samples, and so on. And it's very important to have a control lab, let's say mainly for confirmatory method with high performance level, and to have expertise. I have explained some problem regarding nortestosterone, boldenone, thioracil. You have to keep in mind that it could happen, and to be very careful, mainly if you give a positive result. And the last one is to evaluate the penalties even in positive result. I would like to thank for your attention. To excuse me for my poor English and my French accent, but I hope that you have appreciated this talk. Thank you very much.
Ruba, you are muted, Ruba.
Thank you very much, Dr.
Yes,
Philip. This was really a very beneficial, detailed, technical presentation, and we really appreciate all the content that we have taken.
Thank you, Ruba, but I hope that I was not too long, because I think it was 1 hour and a half.
Great.
Okay.
You were great. I do have some questions, but I would like to start with a question that we had. Is there any benefits from of course, from a scientific point of view, of using orally active steroids in chicken? If yes, do we have any specific example?
No, it's not very interesting, because I don't know by head, but the feed conversion is chicken very good. That's the reason why the chicken meat, the price of the chicken meat is lower than pig and than cattle. And so I think, on my point of view, on industrial condition, I speak on big farmer with a lot of chicken. It's not interesting. It's better to manage all the condition of feed, genetic, and so on. But let's say in Europe, I think that nobody use growth promoter for chicken, and US also. But I don't know everything, Ruben, but I think that they don't use a growth promoter in chicken. It's not interesting.
Okay. Thank you. And I just want to say that, or just to inquire, you know that sometimes in countries they don't accept or they are not registered in this country, some of these beta agonists. You have them like used in some countries. So when we import these products and we test them, even if they are approved by the MRL, still, you know, it is not registered in our country. It is not approved to be used in our country. And then we find limits, even if these limits are acceptable. What do you think the decision should be?
So if you understand well, the question is regarding beta agonists, and you import meat from a third country where beta agonists are allowed.
Yes, that's it.
Yeah, but the question is to know if beta agonist is allowed in your country.
If it's not allowed,
Well, you have to refuse if you can prove that there is. That's the problem that we have, let's say, in Europe with meat imported from US. As you know, okay, in US, natural hormone, trembolone, and so on are authorized, but it's very difficult to prove that they are present in the imported meat. Because first, they respect a withdrawal time. They wait, I don't know, the withdrawal time for each compound. And in the meat, the level of this active compound are very, very low. So the problem is to detect this compound. And there was a lot of discussion in Europe 30, 40 years ago about that problem, but they've decided to not allow. But every year there is import meat from Europe with agreement between US, South America, and Europe, and hormones are not allowed in Europe. But let's say, Ruba, it's a political problem.
Yes, I do. I do agree. There's a question from Esther Adjuzor. Can you hear me?
Yeah. Hello.
Yes. Can you hear me? Okay, it's a raised hand. Anybody would like to ask another question? Okay. So
Ruba, if I may, there are some questions in the open questions that are left.
okay.
Question from Dr. Iman Jassim: how to differentiate between natural and artificial hormones if we detect them in meat samples by LC-MS-MS? I believe you maybe have addressed that, but if you can just follow up.
Yes, but I have explained it's impossible to see the difference by classical LC-MS-MS between endogen and exogen natural hormone. That's the reason why you can use ERMS. So I have explained we make the ratio of carbon, but to be honest, the level of natural hormone in meat are very, very low. So I have a lot of difficulties to imagine that we are allowed to detect the difference from in meat from natural and injected steroid. That's really a problem. We can do eventually in urine or in plasma, but in meat, no. That's the limit of the technique. But you cannot do by classical LC-MS. Forget it.
Okay. Thank you very much. Any more questions?
Hello.
Please, Basil.
Yes. Assalamu alaikum. Thank you, Professor. Thank you, Dr. Roba. I have a question. Actually, I'm not a vet doctor, but according to my knowledge, antibiotics are allowed in accepted limit
Yeah.
to be available in poultry or meat. My question is there antibiotics can be used as well as growth promoters? And how we know which antibiotics which have two functions? Because according to my knowledge as well, growth promoters are not allowed to be presented in the final products of poultry or meat.
Yeah. Your question, if I understand well, is regarding the use of antibiotics as growth promoter.
Yes.
Yes, I have not spoke about this the problem because we also have antibiotics, coccidiostat, and so on. Regarding the residue, I have just limited today to the problem of hormone. But on my knowledge, there is some antibiotics used, and I know better the sector of cattle, like virgiamycin, carbadox, lincomycin, but now they are no more allowed. In fact, these antibiotics, they shift the modification of the flora in human, but it's no more allowed as growth promoter. And these antibiotics are no more commercialized in Europe. I don't know in your country, but I think that some of them are still used in some part of the world. But I have no experience in that field. Excuse me, Basil.
Okay, thank you, Basil. Dr. Philip, there's a question on tilapia. As many growers use 17-alpha methyl testosterone to produce the monosex tilapia fry.
Yeah.
Is your information about that? Also, this contaminated water released to water channels, which used in irrigation, in irrigating and watering animal, besides many leach for groundwater. So this is from Muhammad Sulaiman.
Yeah. I know that it's used for orientation in sex in a fish, but I don't know the concentration that we use for that. And I suppose that if they are released in the water, you have a very large, a very big factor of dilution. So I'm not sure that we can still detect the methyl testosterone in this water. And I cannot imagine that it's still active for other animals.
Thank you very much.
You have a factor of dilution, an incredible factor of dilution, but I don't know the technique. I know that it exists, is for sex orientation in fish, but I don't know the practical aspect of this technique. Sorry.
No worries, doctor. And so before we conclude, I like to extend my sincere thank you to Dr. Philip. Really, this was valuable, and the insights we heard, and very incredible information session. And I'm sure everybody gained lots of information and had like an understanding of the importance of hormonal residue monitoring programs and the role in food safety. So thank you once again, Dr. Philip, for your expert's insight, and to everyone for your participation. And kindly note that the presentation of Dr. Philip will be available soon at G4's website, because we had this question also in the chat box. And dear participants, would like to use this opportunity today to announce our third Global Food Regulatory Symposium taking place next week as part of the 18th Dubai International Food Safety Conference.
Just to finish, Ruba, I would say that if somebody needs some information, do not hesitate to contact me. I have no problem to give you the information and the correct person to help you in this field.
I appreciate that, Dr. Philip. And also looking forward to seeing you all at our
I don't hear you.
Thank you very much, Dr. Philip. And again, I would like to remind everybody of our next webinar, which is going to be on the 29th of this month. And the subject again would be threshold-driven risk management practices for allergen in food. Thank you, everybody, and looking forward to seeing you all again. Have a good day. Thank you, Dr. Philip.
Bye-bye, everyone.
Bye-bye, everybody.
The Global Food Regulatory Science Society. GFORCE mission includes knowledge generation and dissemination, underpinning food regulatory decisions, in support of food control systems. Consistent reliance on food regulatory sciences enables robust food control programs.